Hey everyone, this is Narendra and you're watching to my channel BioGuru. In this video, we are going to learn about the light reaction of photosynthesis. Light reaction includes following events. First event, light absorption. Next, water splitting. It is also called as photolysis. Then release of oxygen. Then formation of ATP and NADPH. NADPH full form nicotine amide adenine dinucleotide phosphate, H here reduced form. Clear? So now all these events we are going to learn, we are going to study under the heading that is photophosphorylation. Right? So what is photophosphorylation? The process of formation of ATP in chloroplast in the presence of sunlight is called as what? Photophosphorylation. Basically, it is the formation of ATP. Formation of ATP is called as phosphorylation here. And where this formation of ATP is taking place? In chloroplast. And that too in presence of sunlight. Hence, this process is called as photophosphorylation. Is that clear? Now, photophosphorylation is of two types, namely non-cyclic photophosphorylation and cyclic photophosphorylation. Now let us have a look at the non-cyclic photophosphorylation. Here you can see LHC, that is light harvesting complex, and this light harvesting complex, it is present in photosystem. Here you can see the both photosystem are present, photosystem 2 and photosystem 1. PS2 absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit further from the nucleus. These electrons are then accepted by an electron acceptor, which sends them to an electron transport system. Then, electron transport system further transfers the electron to the photosystem 1. Electrons in photosystem 1 are simultaneously excited on receiving the wavelength of 700 nanometer. These electrons are again transferred to another electron acceptor having a greater redox potential. From the electron acceptor, electrons are transferred to the molecule, that is NADP+. Addition of these electrons reduces NADP+ to NADPH plus H plus, and this reaction carried out by the enzyme NADP reductase enzyme. So the flow of electrons assumes the shape of Z letter when all electron carriers are placed according to their redox potential. Hence, the process is called as Z scheme. This process is also called as Z scheme. Why? It is because the flow of electron assumes the Z shape when all the electron acceptors are arranged according to their redox potential. Now, the electrons lost by the PS2 do not come back to it. Observe carefully. The electrons, upon receiving the wavelength 680 wavelength, the electrons are transported in a one-way direction and eventually it is received by the NADP+. So the electron which is lost from here is not coming back to it. So there is a non-cyclic transport of electron taking place. So during this process, when the electron passes through the electron transport system, there is a formation of ATP. There is a formation of ATP taking place. Right? Now, the synthesis of ATP in presence of light in chloroplast during non-cyclic transport of electron is called as what? Non-cyclic photophosphorylation. Clear? Now, the question is, when the electrons are lost and they are not coming back to the photosystem, from where the continuous electrons are coming? Who is going to compensate this lost electron? Here, associated with the photosystem 2, there is a one more complex where it splits the water molecule. So that splitting of water molecule is called as photolysis. The electrons which are lost from here, that is photosystem 2, are compensated by the water molecule upon splitting. Right? So here the 2 water molecules are required, where upon splitting it generates 4 protons, 1 oxygen molecule and 4 electrons, and these electrons are taken in photosystem 2 so that it can continuously supply the electrons and generate ATP during non-cyclic transport. Not only ATP, but also there is a formation of NADPH. Clear? Okay. Here the splitting of water taking place and it is associated with the photosystem 2. So this photolysis is also helped by the Mn and Cl, manganese and chloride ion. Clear? So let us have a look at the definition of non-cyclic photophosphorylation once again. Here it is. The synthesis of ATP in presence of light in chloroplast during non-cyclic transport of electron is called as non-cyclic photophosphorylation. Okay, now let us study about the cyclic photophosphorylation. In cyclic photophosphorylation, there is the cyclic movement of electron, and this cyclic photophosphorylation takes place only in photosystem 1. Only photosystem 1 is functional over here. There is an absence of photosystem 2, and this scheme possibly occurring in stroma lamellae, because the stroma lamellae lacks both PS2, that is photosystem 2, and NADP reductase enzyme. So in this cyclic photophosphorylation, only ATP is generated, not NADPH. When the photosystem 1 receives the light, it causes the loss of electron, and this electron is accepted by the electron acceptor. Further, it transports these electrons to the electron transport system. From the electron transport system, once again these electrons are coming back to the photosystem 1, causing the cyclic flow of electrons. So during this cyclic flow of electron, there is a formation of ATP. So the synthesis of ATP in chloroplast in presence of light during cyclic transfer of electron is called cyclic photophosphorylation. Clear? Okay, let us have a look at the points. As I already told you, in this scheme only PS1 is functional, hence the electrons are circulated within the photosystem. This results in a cyclic flow of electrons. This cyclic flow results only in the synthesis of ATP and not of NADPH plus H+. The synthesis of ATP in chloroplast in presence of light during cyclic transfer of electron is called cyclic photophosphorylation. And this scheme could possibly be occurring in stroma lamella because it lacks both PS2 and NADP reductase enzyme. Okay, now let us study about the Chemiosmotic Hypothesis. It is postulated by Peter Michael in 1961. This Chemiosmotic Hypothesis explains how ATP is synthesized in chloroplast. So we have studied that during cyclic and non-cyclic movement of electron, ATP is synthesized. Exactly how it is going to happen, so that is explained by this hypothesis. What is that hypothesis? Chemiosmotic hypothesis. So for the ATP synthesis, it requires membrane, proton gradient, proton pump and ATPase. Let us understand all these things through by diagram. Basically, this diagram is of thylakoid. This is the thylakoid membrane. Inside that there is a space called lumen. Outside there is a stroma. Associated with the thylakoid membrane, there are photosystem 1, electron transport system. This is one of the component of electron transport system, and photosystem 2. So in order to synthesize ATP, it requires proton gradient, the difference in the hydrogen ion across the thylakoid membrane. So how this difference is created, how this proton gradient is created? There should be high concentration of proton inside the lumen compared to that of stroma. So how this proton gradient is created? So there are certain reasons. What are those? Let us see. One of the reason is the photolysis. Photolysis takes place inside the lumen and it is associated with the photosystem too. In photolysis, we know that water molecule is split into hydrogen and oxygen. Oxygen is liberated, whereas the hydrogen which is generated get accumulated into the lumen. And this hydrogen cannot directly pass through the thylakoid membrane. It is because thylakoid membrane is impermeable. It will not allow directly this hydrogen to pass through it. So, because of that reason, the hydrogen ion, that means proton, get accumulated over here. This is the one reason. So, there is one more reason for increase in the concentration of protons inside the lumen. We know that electrons are transported from photosystem 2 to photosystem 1 through electron transport system. So when the electrons are passed through the electron transport system, protons are pumped from stroma to the lumen. So because of this reason, there is a high concentration of proton compared to that of stroma. There is one more reason why there is a high concentration of proton into the lumen, because the NADP reductase enzyme is found towards the stroma where it reduces NADP plus to NADPH. Here the hydrogen is utilized. As a result, the proton in the stroma decreases in number while the protons in lumen increases in number, thus creating a proton gradient across the thylakoid membrane. The proton gradient is broken down. It should be broken down in order to synthesize the ATP. How it is broken down? As I already told, protons cannot directly pass through the thylakoid membrane because they are impermeable to the proton. But still, this gradient should be broken down. The proton gradient is broken down due to the movement of H plus from the lumen towards the stroma through the transmembrane channel. This is a transmembrane channel of CF0 of ATPase. So this is the ATPase particle. It is having two parts. CF0, which is embedded in the thylakoid membrane, and there is one more part which is protruded outside the thylakoid membrane, that is CF1. It is having ATP synthase activity. So it is because of the proton movement, H plus movement from the lumen into the stroma through CF0, it causes the conformational changes, structural changes in the CF0 that results in the formation of ATP from the ADP. Since the formation of ATP is due to the diffusion of the proton by the diffusion force similar to the osmosis. We know that there is a high concentration of proton in the lumen and there is a less concentration of proton in the stroma. So there is a diffusion force similar to the osmosis. So this process is referred to as chemiosmosis. Let us stop here. I hope you found this video useful. If so, hit like button, share this video with your friends, classmates. If your teacher, share this video with your colleagues. If you are not subscribed yet, please consider subscribing. I will see you in my next video. Till then, take care.